Hello and welcome to Maths on the Move, the podcast from plus.maths.org. I'm Marianne Freiberger.
And I'm Rachel Thomas. Today we're continuing our special Euromaths series of podcasts, which all feature interviews that you did, Marianne, at the European Congress of Mathematics that you went to in Seville in the summer. And the podcast series is supported by the London Mathematical Society. Now, before we started the recording, you said this week we're going to talk about topological data analysis. Now, I know what data is, bits of information, and as we know, we deal with a lot of this every day, growing more and more in the era of big data. And I know what topology is. It's a way of describing shapes that allow for deformation. So famously, a coffee cup is topologically the same as a donut because you can stretch and squeeze and pull a donut to form a coffee cup and back again. But Marianne, what is topological data analysis?
Well, I found out about topological data analysis, or TDA for short, from Heather Harrington, who at the European Congress of Maths, she gave the London Mathematical Society lecture, because at every ECM, the LMS hosts a special lecture. Now, Heather is professor of mathematics at Oxford, and she's also a director at the Max Planck Institute of Molecular and Cell Biology and Genetics in Dresden, in Germany, and an honorary professor at the TU in Dresden. And she's the co-director of the Center for Topological Data Analysis. So she's a lot of things.
She's a very busy woman.
She is. Now, to get an idea of what topological data analysis does and why it's really useful, she gave me the example of proteins. Now, proteins can have very complex structures if you think of them as objects sitting in three-dimensional space. And one thing that people are particularly interested in is this thing called protein folding.
I've heard of protein folding. So the chain of amino acids that make up the protein, it kind of folds to give some complex 3D structure sitting in space. So it's no longer just one long line, one long chain of acids. It's kind of folded into this interesting structure. And it's really important, isn't it? Because if it doesn't fold exactly the right way, it can lead to diseases and things like allergies and things like that, isn't it?
Yeah, that's right. And protein folding is a very, very active research topic. So I heard from Heather as well that there's even an AI system called AlphaFold by Google DeepMind, which uses AI to predict the protein's 3D structure from its amino acid sequence. And to make such an AI system, you need lots and lots of data about proteins and the way they fold so that the AI system can learn from that data. So there's lots of research going into protein and its folding.
So I imagine if you're going to do sort of a statistical analysis of all the data you might have about proteins, or to train an AI on that sort of data, you need a really good way of describing the shape of this structure of a protein and the way it sits in space.
Yeah, so that's exactly right. Now, if you think about it, if I showed you a protein, how would you describe the way it looks in the most precise way, in a way that a kind of computer can understand?
Well, my first thought would be to use something like writing down the X, Y, and Z coordinates in 3D space of all the points and the protein structure. I know computers do store 3D shapes in that way. So I can imagine maybe for each, you choose a point on each amino acid, and that would be the point in X of the X, Y, and Z coordinate of that acid in space, and then how they're linked together. But that's quite a brute force way of describing something.
Yeah, so you could do that. Like for any shape, you could describe it by giving the X, Y, Z coordinates of every point in it. But then one problem with that, apart from that you have a long list of coordinates, is that, what if you got the coordinates slightly wrong because they're difficult to measure, for example? Then you might accidentally end up describing a completely different protein, or maybe one that doesn't even exist, or maybe you miss a crucial feature of the structure.
So I suppose that's where topology could come in. Um, you might want some kind of descriptor of the protein, some way of describing it that's somehow kind of tolerant to a bit of deformation, a bit of mistake, a bit of noise, that although there might be this slight error, but it still captures the important structural topological features of the shape, such as whether the structure is made up of several disconnected bits, or whether it contains a ring or a loop or lots of branches.
Yeah, exactly. So topological data analysis is about developing such descriptors for data. So the data could be proteins. And what you want is a descriptor of this data that is kind of tolerant to a bit of information and captures important feature. Here's Heather explaining it in her own words.
So the idea is that exactly what you said, which is that you have some points in three-dimensional space, like the carbon atoms in the protein structure. And so you have XYZ coordinates. And then maybe you don't want to only study the geometry, you want also some of the topology will explain. So TDA uses topological invariance, but you extract some geometric information as well, because you're really looking at this as a multi-scale descriptor. And so if your input data, if it is perturbed just a little bit, the protein is perturbed just a tiny bit, which we know proteins are not only rigid, right? They are dynamic. So if it perturbs just a little bit, you want to get a similar descriptor out, and that's one of the strengths in TDA. And so we looked at, and this was actually with the undergraduate students, so it was one of these undergraduate research projects where they took protein data and then ran this topological pipeline where you compute different topological features in different dimensions, so like how connected or some loops, so one-dimensional holes. And from that, they could kind of get a fingerprint or a descriptor of different proteins. And what we found is that proteins that are similar, cluster together, are very similar in the topological kind of fingerprint, the output of this.
So Heather mentioned a pipeline. What does she mean by that?
So as she described in her talk, there's a whole pipeline of mathematical things you can do to raw data to at the end come out with a fingerprint describing that data. And this pipeline involves this very interesting thing that is called a filtration. Now, Rachel, in order to describe or give an idea of what this filtration is, let's start by imagining something, right? So
Okay.
I want you to imagine that the object that we're trying to describe is just a bunch of points, right?
Like in 3D space, points in 3D
In the plane,
space?
they can be in the plane.
Points in a plane.
Yeah, let's imagine they all are arranged in a sort of circular fashion. They sit on a circle, right?
Okay, so I'm imagining kind of a blank piece of paper with dots
Yes.
forming into a kind of circle.
Yeah.
Okay.
And imagine, let's say that there's around 20, let's say there are 20 points. And let's say they all have equal distances between them, let's say distance of a centimeter. Okay. Right. Now imagine if I draw a tiny little circle of 1 millimeter radius around each of these points. What do you see then?
Well, I now see a ring of kind of circles, but they're not going to, the circles are going to be distinct from each other because the points are kind of separated by a bigger distance than the radius of the little circles. So it's like a bigger circle which is made up of little circles. Yeah?
Exactly. That's what you see. So it's a bunch of 20 little discs if we take the inside of the circle as well, right? And they don't overlap, as you say. Now, what if I gradually increase the radius of these circles and keep going? What will happen eventually?
Well, eventually the discs, the areas inside these circles will overlap each other as they get big enough.
Exactly. And what do you see once they've started overlapping?
Then you kind of see a continuous circle.
Yeah, exactly. You see sort of like a ring type structure as they've started to overlap. So you have a ring with a hole in the middle, right?
Yeah.
Okay, now what if you keep increasing the radius of these circles if you make them just really, really big?
If you make them really, really big, then eventually they're going to cover not just the ring, but they'll end up covering all the way to the center of the ring. So you'll end up getting kind of like a big disc, a big interior of a circle covered, which is made up by all these individual circles.
Yeah, exactly. So what happens is that all this ring of circles suddenly starts merging together in the middle, and you get like a blob made up of exactly. So that's what you get. So this process of increasing the radius of the little disks, or if we were in 3D space, they would be balls. It tells us something about the configuration of the bunch of points, right? Because it tells us that initially they're separated because we get lots of individual disks. The fact that they join up to form a ring means they're arranged in a kind of circular fashion. And then eventually it just all merges up into a blob.
So the kind of characteristics of the shapes that are created along the way of the increasing in size of these disks gives you some sense of the formation of that data that you started with.
Yeah, exactly. And this is what the filtration is. And more generally, a filtration involves increasing a relevant parameter. In this case, it was the radius, to capture information with a little bit, you can do that with a little bit more mathematical rigor than we have just let on. Here is Heather explaining it.
Exactly. So you have a filtration is looking at each value of this parameter, which could be the radius, and then looking at what is the shape that results at each parameter value. And that shape we want to quantify, and there's a topological invariant that's computable called homology, where we can extract the connectedness in the shape of that data through this union of balls.
Well, I really like this idea of filtration. It's a very neat idea. And I like the fact that it's sort of robust to little changes. So if some of the points in our example were moved a little bit, the overall nature of what's happening during the process, the way the disks join up first into a ring and then into a blob, that wouldn't massively change.
Yeah, that's true. And from such a filtration in topological data analysis, you can build what the topological data analysts call a barcode, which describes the structure you're interested in. And that's the kind of fingerprint that you're looking for to describe the structure.
So now I have a picture in my head of topological data analysis scanning these things at a self-checkout.
Yeah, or it's actually producing the barcode that you can then any statistical analysis will then scan all these barcodes and kind of process them. So yeah.
Kind of identifies the object that you're looking at, the structure you're interested in.
Exactly. So here's Heather describing this idea of a barcode a little better.
So at any point in if we're thinking of radius, right? If at any radius value, any parameter value, the number of bars, it tells you the number of topological features in that dimension. So when you're thinking of dimension 0, this is connected components, so clusters in data. And if you're thinking of degree 1, this is the number of kind of loops in your data. And because you are this barcode, the left and the right endpoints of each interval are telling you when a feature is born and when a feature and when a feature dies, then the length of that gives you a measure of persistence. So how the size of that feature, do you have a big loop that will last a long time or not?
Wow. So what would the barcode look like, for example, of that 20-point data set we were imagining before, sort of arranged into a circle?
Okay, I'll try to describe it. So let's see if I can do this and it makes sense. So first of all, imagine putting down a ruler on a piece of paper just horizontally. Because is
this the piece of paper with the dots on it?
No, it's a separate one. It's the one that you're going to draw the barcode on.
Right. Okay. So now I've got a separate piece of paper and I'm putting a ruler down on it. Yeah.
Horizontally, because the barcodes we're used to from products that we buy, they're all vertical lines. But this barcode will be made of horizontal lines. So you put Okay. Now, above the bit of the ruler between 0 and half a centimeter, we draw 20 parallel lines, all stacked on top of each other.
Is that because we've got 20 points of data?
It's because as long as the radius of the little discs, as long as that's less than half a centimeter, they don't overlap, right?
So you've got 20 separate parts when you've got half centimeter circles. Got you.
Exactly. So you've got this stack of 20 horizontal lines sitting on top of each other, indicating that we have 20 connected components, as they're called. And we'll give these 20 lines a color as well. Let's make them red. And this indicates that topologically they are disks. So they are things that don't have holes in them.
So the color indicates the structure that the lines represent.
Yeah, so it's the topological structure, which is a disk, something without a hole in the middle. Now, to the right of that stack, which would then if you go back down to the ruler, correspond to values of the radius of the circle being bigger than a half. Okay? We draw a single horizontal line and we will color that green.
Okay.
So this indicates that as soon as the radius is bigger than a half a centimeter, because the points are 1 centimeter apart, so if around 2 points there's a circle of a radius of bigger than a half, then they will overlap, right?
Yeah.
So as soon as the radius is bigger than a half, what you see is no longer 20 disconnected disks, but a single ring.
Yeah.
Right? So that's why we only draw one horizontal line.
And it's a different color because now it's a ring rather than a disk.
Exactly. So now topologically, a ring and a disk are very, very different because one has a hole and one doesn't. So we indicate this by color. And now, of course, we know that as the radius increases further, at some point, the ring disappears and turns into a blob. And this will indicate
topologically the same as a disk.
Exactly. So that will be indicated in our barcode by the fact that this green line has also a finite length. It stops at some point. And then what we get for values of the radius bigger than that point where the ring disappears, we will again have a single red line. And that indicates that there's now a single simply connected bit without holes in it.
Have we got a picture of this? It'd be great to put it in the show notes.
We will make a picture of it and put it in the show notes. I mean, this is sort of the rough idea of what the barcode code looks like and how it encodes how these different topological features appear and disappear as the radius increases.
And it's quite a clever way of encoding both the count of how many components you've got, the color of how many, indicating what type of components they are, and then this positioning of the lines indicating what scale you're sort of looking at. It's really interesting.
Yes, and as Heather already said, it's sort of this barcode idea is robust to small changes. So if you moved the little points a little bit, the barcode would look still roughly the same. It would only be slightly different. And I mean, this that there are mathematical results which prove the kind of robustness of this approach to characterizing these shapes that we were looking at. And what we just described is, of course, a very simple example of where the object we wanted to characterize is just 20 points sitting in the plane. But with the power of mathematics, you can do something sort of similar for much more complex data than just bunches of points. So, for example, you can create barcodes for data that live in higher dimensional spaces that we can't even visualize.
So again, that's that illustration of the power of maths, that you can take mathematical ideas for things that we can really easily visualize, disks, rings, expanding circles in 2D, and you can just extend those ideas mathematically to cover higher dimensional situations. It's amazing. So what kind of things do people do with topological data analysis?
That's what I asked Heather.
So there's been, there's an entire database called Donut, which is all examples of data sets that have been useful with TDA. And so many of the problems that I'm motivated to study are from biological sciences. But in the field of TDA, there's been a lot of success looking at neurotopology and neuroscience because the brain is so complicated, trying to kind of integrate information. This gives you a way to integrate it in some way and study across different scales or different weights of connections in the brain. I've been focused a lot in disease, so it turns out that, you know, if someone is unwell, they'll take some type of samples. It could be a tissue sample, for example. And from the tissue sample, a pathologist or some type of machine learning algorithm will say these are the cell locations and identify what types of cells they are. And so we have points in the plane of what different cells are. And then one question is, are there certain patterns that are, you know, kind of characteristic of certain diseases or progression to a disease? And so one example is cancer. There's different stages often in cancer, and same thing with treatment in cancer. And so we have, over the last few years, applied quite successfully in building new TDA approaches that's really been motivated by these types of problems. So can we describe, quantify the patterns of certain spatial, certain cells when we are given that spatial information? And usually these are some type of immune cells, so some in subsets, so like T cells or macrophages or I don't know, many different types of cells. And so that's given them a way to quantify that, and we've made some predictions. So yesterday I discussed this, is very excited in a project of, it was mouse kidney, and with collaborators in Oxford, and we made a prediction that there's a ring with the topological data analysis, that there's a ring of immune cells, which then they experimentally validated, and we were able to use the, you know, actually fairly sophisticated recent topological data analysis was the multi-parameter persistent homology, which is mathematically really challenging, but there's no, as I said, there's no complete invariant. There's only incomplete invariance, but still we can make these hypotheses and actually test them. So we found some new biology.
So this is maths helping to discover new biology.
Yeah, it's amazing, isn't it? I asked Heather to explain us a little more about this.
So I think one way to think about mathematics is that we often build from the ground up, the foundation, right? And everything is kind of built on that. So you have pieces of a puzzle, or, you know, blocks, and you build up your building with these blocks. And in biology, you don't have all the pieces. So it's kind of, you can't see all the pieces to build that. And so I think in that sense, they're still trying to discover what are all the pieces, and can math help kind of identify what those pieces are. And that's what I think mathematics can really, help uncover so much. I think what's changed a lot in biology is now measurements are taken, like I mentioned, this example of a tissue sample, but they have, you know, the genomic information from the tissue sample, they have the image, then they have all the information about maybe that patient and like, I don't know, other, right? So they have many different types of data, and all about that one sample at that one point in time, for example. And I think they just, biology and medicine need new approaches. Yeah. That can bring it all together, that are rigorous, that are stable to perturbations, that, and it's still pieces of a puzzle, right? But I think mathematics can unify and simplify things, or at least that's what we try to do, and then have some insight, so have it be interpretable. And that's one thing in topological data analysis, that there's a lot of interpretability and stability. So it brings, I think biology is kind of a playground of many problems that we're trying to piece together, pieces of the puzzle that we can't even see.
So topological data analysis, it's quite a new thing by the sound of it. So what kind of things have been happening?
Well, lots of things, really. In the UK specifically, there has been this thing called the Centre for Topological Data Analysis that Heather is a co-director of. And she told me a little more about that.
Right. So I think the UK is very progressive, and the EPSRC has been very good at kind of these interdisciplinary, whether it's interdisciplinary within mathematics or within the sciences, to support this. And so this was an application where they had a call for new approaches to data science. And so we had a couple small workshops on topological data analysis, and had an incredible number of people, even from industry. I think at one point we had an industry event with over 50 different people that came, saying, "Oh, what's going on with TDA?" So we applied for the center grant, and then we started it in 2018. And it's been really wonderful. We have over 50 members, so something like 11 faculty, and then many industry partners, and it's been across 3 or 4 nodes, depending, because some people moved, how you count. But it's been this really wonderful way to have a community, and the advances mathematically have been phenomenal. It's just such an active field worldwide. It's really an active field, but I think having the center brought a critical mass to the UK and many new positions. So we developed lots of new theory, mathematical theory, but also, as I said, it's application driven, so new insights for us in Oxford, we focused mostly on biomedicine, but then in Liverpool, they were looking at materials, and in Swansea and Durham, more physics problems. So, each of kind of the different people bring what their interests and their expertise are, and it was really a nice mix, and we had regular meetings to continue informing each other and discussing rapid advances in the field. So it's been really great, and a lot of energy, like positive energy, I would say.
It's so interesting to hear about, such a new and so very active area of research.
Yeah, it's been really fascinating, because that was the first time for me as well to find out about topological data analysis. So we'll see. I'm sure we'll hear lots more about it in the years to come.
So that's it for today's podcast. We hope you've enjoyed finding out about topological data analysis. And if you've enjoyed the podcast, please do recommend us to a friend, or rate and review us in your podcast app. It helps other people find us. But that's it for today. Thanks for listening, and bye for now.